More experience equals bigger brain is a psychological experiment reading which was testing brain changes from experiences. The reasoning that led to tests on brain development is from ideas such as parents thinking a child needs constant stimulation for proper development. This line of thinking led to questions asking if physical changes of the brain are affected by experiences. Does experiencing more increase the size of our brains? And if so, what is actually physically changing in our brains? In 1785, an anatomist named Vincenzo Malacarne tested animal experience and brain development. He would use 2 dogs and 2 birds of the same litter and train only one of each. The other would be left untrained. Once the animals died, Malacarne dissected their brains and noticed that there were in fact physical changes in the trained animal's brain that were different from the untrained. Studies regarding brain development were put off for a long time until questions arose of whether head size changes could be in relation to brain development changes. It wasn't until almost 200 years later that another study was done that may have justified or discredited this. The new test was conducted by Mark Rosenweig.
Through this experiment, rats are to be used as test subjects by scientists to measure the brain and its forms. Rats are being used, more likely so because they are easier to care for and smaller in size, less expensive, and mainly because the rat's brain is smoother than most higher animals. Also, rat strains can become inbred, allowing for researchers to look at their genetics if they so desire. The hypothesis for this research was that animals that are raised in highly stimulating environments will show greater differences in brain growth and chemistry when compared to animals reared in plain or dull circumstances.
3 male rats were chosen from each litter. They were then randomly assigned to one of the 3 conditions. 1 rat remained in the laboratory cage with the rest of the colony. Another was assigned to what Rosenweig termed the enriched environment cage, and the 3rd was assigned to the impoverished cage. Remember, 12 rats were placed in each of these conditions for each of the 16 experiments. The 3 environments were the standard laboratory colony cage, which contained several rats in an adequate space with food and water. The 2nd was the impoverished environment, which was a slightly smaller cage isolated in a separate room with 1 rat placed alone with adequate food. And the 3rd was the enriched environment, which was virtually a rat's Disneyland. 6 to 8 rats lived in a large cage, furnished with a variety of objects which they could play with. The rats were allowed to live in these different environments for various periods of time, ranging from 4 to 10 weeks. The rats' brains were then measured, weighed, and analyzed to determine the amount of cell growth and levels of neurotransmitter activities. In this latter measurement, 1 brain enzyme was of particular interest. Acetylcholine lysoterazio. This chemical is important because it allows for faster and more efficient transmission of impulses among brain cells.
Results indicated that the brain of the enriched rats were indeed different from those of the impoverished rats in many ways. The cerebral cortex of the enriched rats was significantly heavier and thicker. Also, greater activity of the nervous system enzyme acetylcholinesterase mentioned previously was found in the brain tissue of the rats with the enriched experience. Although no significant differences were found between the 2 groups of rats in the number of neurons, the enriched environment produced larger neurons. Rosenzweig also found that the ratio of RNA to DNA was greater for the enriched rats, meaning that there were higher levels of chemical activity in the brain. Rosenzweig and his colleagues were confident that even though the differences in environment were not large, they are genuine. When the experiment was replicated, the same pattern of differences were found consistently. They observed that the weight of the cortex increased in response to experience. Under great magnification using the electron microscope, the researchers also found that the synapses of the enriched rats' brains were 50% larger than those of the impoverished rats, potentially allowing for increased brain activity.
The significance of this experiment is that its results can contribute to the different studies made by scientists today, like the study of memory loss. There are certain factors, such as experience, that can cause the brain to change. These changes help us understand how our brain develops and will help future research find different ways that memory loss can be prevented. Also, the connection between malnutrition and the knowledge gained during the experiment can help us understand that the lack of nutrients can affect the way our brain receives information and its developmental growth. This growth can be influenced depending on which environment we are placed or developed in.